So this research study took place in Italy, more precisely in Puglia. If you think of Italy as a boot, Puglia is the heel of the boot. What was the situation? That Italian law has a no-kill policy, and stray dogs are captured, neutered, and taken to shelter. In Puglia, we have a problem with stray dogs, so the law permits the release of neutered dog as an alternative to shelter. No, it doesn't. Yeah. So what's the situation? That we have 4 million humans in Puglia, and we have 700,000 dogs. Yeah. So more precisely, 470,000 has an owner, but many of them are free of roaming. And 40,000 are taken in 120 shelters, and 150,000 are stray dogs. So what happened? That every year we have 50,000 newborn puppies, and we have 31,000 babies. So it means that each baby should take care of 2 puppies. So the problem is when a family has 2 more babies. So each family should take care of at least 4 puppies. So as long as the birth rates of dogs are so high, of course, there will never be enough space and homes for all of them. Let me underline that the word stray is not really useful when you talk about population control and management. So it would be better to talk about confined dogs or free-roaming dogs. It is important to manage the dog population as a whole, not just considering stray alone. That's why 46% of roaming dogs are actually owned dogs, and many of them are allowed to roam and mate freely. So controlling reproduction to companion animals is essential to achieve effective population management. Non-surgical methods of sterilization, and I think calcium chloride, could be better accepted by owned dogs, by owners, and so yield a positive impact on canine overpopulation. Okay, our research was about the injection of calcium chloride in the testicles, so intratesticular or intraepididymal. We inject the head of the epididymis. And so we wanted to figure out which one is better in stray dogs or in owned dogs. We used a very large sample of animals. There were 148, and they were divided in 4 equal groups. So 2 of them receive an intratesticular injection, Group A with calcium chloride, Group C with saline solution. The 2 other groups received an epididymal injection, Group B with calcium chloride, Group D with saline solution. Before the injection, dogs were lightly sedated, and all of them were owned dogs. So this is our experimental protocol in brief. So we performed the injection at baseline. Then we performed blood testosterone, and we made sperm analysis at baseline 3, 6, and 9 months later. Contrast-enhanced ultrasound were performed at baseline and 5 months later. At the end of the study, that was a 9-month study, we castrate the dog and perform histology. This is what we inject. The formula was made of 20 grams of calcium chloride dihydrate powder brought to a final volume of 100 milliliters of 95% ethanol, which is alcohol, mixed and sterilized in Falcon tubes. In the pictures on the right, you see all you need to perform the injection. So there's a skin disinfectant, and then we have a cotton gauze, Falcon tube with calcium chloride, and 2 syringes, each dog's. And also there's the famous caliper. The caliper it was needed to measure the testicular width. So, as you can see from the table on the left, those are the dose for the testicular injection we used, according to the testicular width. In the table on the right, there is the epididymal dose we use for, of course, intraepididymal injections. And please note that this is just the 25% of the testicular dose we've been used. That's for anatomical reason, as the epididymal volume is around 25% of the testicular. Tell me the video is gonna work, please. No? Yeah. Okay. This is the video how we perform the intratesticular injection. As you can see, you just need to hold the testicle with one hand, and with the other hand, you perform the injection. We stop injecting and release the pressure with the other hand and remove the needle. That's it. In the other video, the other one, please. You see just a different way to hold the testicle while performing the injection. We found that there is no difference in the way you hold testicle. Just you have to hold it, of course. Okay. This is how we perform the epididymal injection. It was a little bit more tricky because two operators are needed, and also an ultrasound guided is needed. So one operator has the probe and locate the epididymal head. There should be another video here. So the guy with the operators with the ultrasound probe indicate the other one where is the time to perform the injection. It's a little bit tricky because, it's three hands in a very small place. And yeah, that's it. It's the same way as the testicular ones. You just perform the injection and remove. Okay. So what we got? All animals in the study tolerate the injection of calcium chloride. We had no side effect. And at least 24 hours following injections here, and continuing for the first 3-4 days, we have just a slight increase in firmness. But the increase in firmness was a little bit more evident in intratesticular injected dogs, and was also here in group B, which is intraepididymal injected dogs. The firmness was focused on, of course, in the epididymal area. And the same happened with group C and group D control. We have also here an increase in firmness. But at 3 months, the testes became atrophied in group of intratesticularly injected dog. That's it. How about semen evaluation? We perform it computerized, which means that, of course, we have objective results, not subjective results. So they are, quite important. And what we have, that dogs of group A, again, testicular injected dogs, were azoospermic, still azoospermic at 9 months. And dogs of group B were still azoospermic even at 9 months. Let's see more deeply what does it mean. So if you look at sperm concentration, total sperm motility, and progressive motility, in dog of group A, testicular and intraepididymal injected dogs, we got 0. It means, of course, that we had no spermatozoa. That's where, of course, they were azoospermic. So, while if we look at serum testosterone concentration, we had a drop here. Can you? Okay. We had a drop here. It was a statistically different with a statistically different meaning. I mean, it's not zero, but this is a drop. So what we have here, it's more clear. So intratesticular injected dog, as you can see from the blue line, has a drop in testosterone, but we also have a decrease in sexual behavior and aggression. We know that aggression is not linked to testosterone, but this is what we found. Group B, epididymal injected dog, has no drop in testosterone and serum level and no changes in sexual behavior. The same happened in control groups. Okay, what is CEUS? CEUS is a contrast-enhanced ultrasound. It's an ultrasound with the use of a medium injected directly into the bloodstream, consisting in microbubbles. Microbubbles allows the echo-amplification of the structure and reach testicular parenchyma in about 15 seconds. So why we use it? Because we want to characterize testicular abnormalities and damage made by calcium chloride. No, the other one. Okay. I think it works. Yeah, okay, great. Here in the picture on the left, we saw at baseline how testicular parenchyma appears. So this is microbubbles, and this is the tissue highlighted by them. After 5 months, here, in the picture on the right, there is no more structure highlighted by microbubbles. In the normal ultrasound, you see here there is no more tissue in this area, black area. This suggests, of course, a damage. How about epididymal injection? At baseline, you see the coiled tubes of the epididymus, perfectly clear at baseline. And after 5 months, we are not able to see the normal structure of epididymus. So also here, there's the suggestion of a damage. This damage actually was highlighted by the histology. So in the epididymus, there were no sperm cell and there were necrosis. Moreover, we found there's a damage here in this part here. So the damage was not just focused on the epididymal head, but also here in the testicular area. This is the place where sperms move to the epididymus. So a damage here is, something that add value at the damage, of course. On the contrary, not on the contrary, but histology on the testicular side also showed that we have no sperm cells and parenchymal degeneration. So this no, I want to go backwards. Okay, this suggests a permanent damage. Okay, in conclusion, what can we say? That injection in the epididymis was very challenging due to the small anatomical dimension and flexibility. It's better if echographically guided injection and of course needs more than one operator. Sterility was achieved for at last 9 months, but histological damage suggests that we have a permanent damage. We had no drop in the testosterone level, and we can say that it is as time-consuming as a quick orchectomy. So we believe, for all of these reasons, that maybe the optimal use is in owned dogs, where anatomically integrity and testosteronemia maintenance can induce, of course, reluctant owners to sterilize their animals. While intratesticular injection is very easily to be performed. Sterility was achieved for 9 months, but of course also here we think it's permanent. we found that we had a reduced sexual behavior, and we think that it fulfilled the principal requirements for application to a stray population canine. So we think that at the end, the use of both methods together in stray and owned dogs may help fight the global problem of overpopulation. Okay, I would like to thank Parsemus Foundation, Elaine Lisner, and Linda Brand, which is here, for the huge help in scientific, linguistic, and statistic support. My professor, Giovanni Lacalandra, which is here, and Professor Giulia Aiudi, and at the end, Joyce Briggs and Valerie Benka for the patience they show me in many communication we had by email. Thank you.